Hello, 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 hello. I am John Beckmann, Professor John Beckmann, Dr. John Beckmann, the father of Sid, the transgenic mother of sin, your hostess with the mostest. Get a green screen in the back. Hopefully it doesn't turn black. I drink my coffee black. Have my brain tied behind my back just to make it fair. This morning I was in the bathroom and something smelled a little siffy. If it smells siffy, stop taking a whiffy. Was it the 2x1 cup model? What does my fortune hold for this year? Ooh, that's a good one. The road to glory will be rocky but fulfilling. That is dominant. I'm keeping this. Got the inhaler. Is it? I don't know. I feel I have a lot of energy this morning. I don't know if it's because I'm on the third cup. Wolbachia, shut the fuck up and give them species names. Is it because I'm on my third cup, or is it my inhaler? You. I don't know. I got a lot of energy this morning. All right. So I read this paper as a preprint, but I don't want to do reactions to preprints because I don't want to do reactions to preprints because I don't want to be putting things out there that aren't in a final state or reacting to things that aren't in a final state. So I prefer not to react to preprints. So I'm glad I waited for this thing to come out. It's out in Nature Microbiology. I have been kind of thinking about it and talking about it a lot. I've never met Flaminia. So Flaminia Catarrhuccia is the PI on this paper. Kelsey Adams, first author, is a co-first authorship. It looks like, I don't know if there's a co-first authorship or not, but a few authors on this paper. I've never met Flaminia, but I did apply as a faculty member in their department. This is actually my Ask Professor story. And when I was on Periscope, I was tantalizing people by saying, if I want to hit this many subscribers, I'll tell you the Ask Professor story. My Ask Professor story, I can actually tell that now. It was when I was applying for a job. I think it was like a microbiome something professor at Harvard in the department of, I don't know if it was immunology, something, or microbiology. I can't remember what department it was, but it was in Flaminia's department. And I worked on all my letters of recommendation. I worked on all my documents, my application documents. And then when I sent it in, I put an abbreviation for assistant professor, and I was not even thinking about it. I was just put the abbreviation, and for some reason I had like a brain fart, and I put ASS dot, because like as would be weird, and I didn't even think about it. And then because I thought that's what it would be, I think the actual abbreviation for assistant is ASST dot. Obviously, to avoid this reason that when you send this in, then it said Ask Professor. So I applied for the Ask Professor position when I sent in all my application materials, and I did not get that job. Their loss. Harvard was not into the Ask Professor, but Alabama was. I don't think I would fit in very well in Boston anyway. It would have been cool. Definitely, like, everybody has a dream, right? It would have been cool to interview at least for a professorship at like Harvard or a big school like that. But I definitely don't think I would fit in very well. With 6 kids in Boston as an assistant professor, you'd probably wouldn't even make enough money to like get a proper house. So I'm happy to have more space down here in the South. But anyway, it's interesting. What's interesting is that people have been asking me to react to this paper. People have been asking me to take a look at this and give out my thoughts, which is interesting, right? Because they want the old boy professor from Alabama to react to the Harvard paper, which is funny. So I'm going to reread this and see if I can coalesce my thoughts and just put some cherries on top of this and see if I can put it in its context in the CI field. Good to be back. Happy New Year. I hope everybody has a very productive, positive New Year. Oh, so the title of the paper is Wolbachia CidB induces cytoplasmic incompatibility in the malaria mosquito vector. I did not review this paper, although would you believe me even if I said I did or I did not? I don't know. I don't know if people would believe me. I usually don't go out of my way to lie. I did not review this paper, but I do think that's interesting, right? Like that. I did wonder why I wasn't recruited to review this paper, because if you're looking for an expert on transgenics, mosquitoes, and CI mechanisms, there isn't really anybody else in the world. There's nobody else that's got the ring, the CidB ring. That CidB ring. That's a gold CidB ring, baby. So I did wonder why I wasn't recruited to review it. But hey, actually, I like not reviewing papers. That's actually the best. So I actually encourage everybody in the field, everybody in the CI field, please exclude me from reviewing your articles, because I do not have time to review them. And I don't want to review them. So please exclude me. Every time I review a paper, I lose 6 to 8 hours of my life. Please exclude me. All right. So here's the context of this paper. This paper comes out after many studies have now been studying the CI genes in Drosophila. And the new technique, the new thing that this paper does is in the past, people were doing insertions of either full operons or individual genes. So we're talking about the CI factors. We'll call them, some people call them CIFs, some call them by another name. We'll call them CIFs for now. Some people are doing transgenic insertions of the whole operon. Some people are doing transgenic insertions of individual components. Okay. So these are essentially like different types of constructs you'll see in the literature. And most of these studies had been, or actually all of these studies had been in Drosophila. So studying effects of, if you take these CI factor transgenes, you put them in Drosophila under different contexts, make different mutations, etc. What happens to the reproductive phenotypes? This is the first paper to take CI transgenes and put them into mosquitoes, according to the abstract. And they're putting them into Anopheles gambiae, and they're putting them into Anopheles gambiae. Why is this important? The Anopheles mosquitoes are the mosquitoes that spread malaria. And traditionally, Anopheles are not viewed to be infected with Wolbachia, although there are some studies that go out and find Wolbachia infections. I have not gone into that literature. I'm not going to discuss that literature. But essentially, like, one of the big contexts of the field was people had been studying Wolbachia and CI and all these different things, the virus suppression by Wolbachia, but most of these have only been applied in Aedes mosquitoes, which spread more of the viruses. So like Zika, dengue, things like that. Most of these techniques have been applied in Aedes mosquitoes, which are the black and white tiger mosquitoes, because you can find like Aedes albopictus, which has natural Wolbachia infections. So they don't seem to be recalcitrant to Wolbachia infections. And Aedes aegypti, we got lucky in the sense that Aedes aegypti was not infected in the wild, and then you infect it with this heterologous Wolbachia strain WML, and it kind of like is not used to it, and it kind of like boosts its immune system, and it can cause all these beneficial effects that lower vector competence. So most of the Wolbachia research has been applied in 2 different mosquitoes, Aedes mosquitoes and Culex mosquitoes. Okay. So it's always been kind of a hole or a gap in the field, which people have always proposed, which was, can we apply any of this Wolbachia techniques to the Anopheles mosquito, which is the big deal that's spreading malaria, killing lots of people by malaria. So the problem is people had been trying to transfect Anopheles with Wolbachia infections, and that never really quite worked the best. And so there's always been kind of an inability to apply Wolbachia in Anopheles gambiae. Although I think Anopheles stephensi, I think Wolbachia has worked in Anopheles stephensi. Yeah. Okay, I got that right. So here's Zhongji Science 2013. So Zhongji is if you watch my other video, SIT and IIT, Zhongji is a big researcher in Wolbachia and collaborating between the United States and China. So here's a big paper, 2013, Wolbachia invades Anopheles defensive population. So this would have been a heterologous Wolbachia infection. So essentially, like Wolbachia techniques or Wolbachia research has been starting to be applied in Anopheles mosquitoes for, I guess, now the last 10 years, the last decade. But before that, it was kind of like a hole. And so people have been trying to more and more to apply these techniques to Anopheles, and there's been encountering problems. Some things about it have been more difficult than others. And typically the success story that you hear is with Aedes aegypti or Culex if you go way back in the literature. Anyway, so Anopheles gambiae is a big deal. It's a big deal, kills lots of people with malaria. And so the idea is, would any of these tools, these transgenic tools, have any applicability in Anopheles gambiae? And in order to demonstrate that they do have this crossover tool technology, you would have to construct the transgenic mosquitoes, insert the transgenes, and see that they cause CI. So there's an importance, some of the importance of this paper. Let's talk about some of the importance or significance. Significance of this paper is it shows you that these tools are cross-compatible. The transgenic CI tools are cross-compatible. You would be able to use them for sterile insect technique. You would be able to use them in theory for gene drive if you can get it to work in a transgenic alone system. So they're cross-compatible, especially with respect to the worst malaria mosquito, the one that spreads Plasmodium falciparum. So the technologies are malaria applicable for P. falciparum or falciparum. And there's also some mechanistic insights, okay? Mechanistic insights. If you watch the last video about the structures, the SIN structures and the SID structures in PNAS that was just published, I did a little bit of a sidetrack and a discussion on some of the mechanisms. And if you watch my longer video on CI, it's like an hour and 60 minutes or something like that. It's almost like 2 hours long. If you watch that other video, I think it's called CI Stories, Advanced Discussions. There's a lot of mechanistic discussion in that too. And so this is an area of debate in the field right now, is how does CI work? What is it specifically? What are its mechanisms? And this paper provides some interesting mechanistic insights in the mosquito, which is probably, in some senses, you might say it's more reliable than Drosophila, because in Drosophila, many of these studies are taking CI operons. For instance, like our studies, we're taking CI operons from Culex and putting them into Drosophila. So it's a different context. Other CI studies are taking Drosophila transgenes and putting them in Drosophila. So that's a good context. But in some senses, moving these Culex genes back into a mosquito, Anopheles, and looking at the mechanistic insights, it might be more reliable in the mosquito setting than they are in the Drosophila setting. So it's a really interesting paper that is providing mechanistic insights into how these operons work in a mosquito setting. Okay, so that's the backdrop. That's the importance of significance, according to my opinion, after now reading the abstract. And it looks like the paper is specifically mechanistically, it looks like it's specifically picking a bone with the 2x1 cup model. I'm going to get in trouble. I'm going to get fired someday. Hey, I'm doing a movie right now. But do you need to be in here? What's up? Yeah. Is that okay? Or are you going to do like microinjection stuff? No, you're good. You're good. I can edit all this out anyway. I'm reading this paper that just came out. You definitely got to get that fixed, because you got to get that swipe access. Which one? Nature Micro one? People have been asking me to react to this preprint. So I've been like aware of the paper for a long time. I read it a while ago. But I don't want to react to a preprint because it's gonna get changed through peer review. You know what I'm saying? So I was waiting to react to it until it was published, and then it finally came out in Nature Micro. Shayun let me know that it came out. Like I knew it was coming out. Like I knew they were gonna get it through, because I always hear like rumors through the Wolbachia dark web. Oh, I just described that. the special part about this paper is this is the first paper to like do transgenics of the CI genes in actual mosquitoes, and they do it in the malaria mosquito, which is kind of a big deal. So that's one of the big pluses of it, and they have some interesting mechanistic insights which support our hypothesis as opposed to the other hypothesis. So that's another reason I really like this paper. Oh yeah, some people would say that that's biased, but I don't know. Like, what? No, I didn't. I did. Well, yeah. Like, I didn't review it. In terms of the paper, but yeah, people were emailing me and like asking me to like make a reaction or like give my thoughts on it for sure. Like, and then multiple people like emailed me and like, hey, did you see this? Like, what do you think about this? Yeah, it just recently popped. I think it popped in at the end of last year, 2021. All right, yeah, I get self-conscious when there's other people in the room. Okay, again, like if you don't know about these models, then you're not up to date in the CI field. So I don't want to go like I don't want to spend another 30 minutes talking about models. But 2x1 is essentially the two genes in the operon are essential and required for induction. Again, this is very nuanced, but one of the discrepancies is there's other models, a TA model, which is strict TA model implies induction would be by the toxin and rescue by antidote. I hate doing this every fucking time. Essentially, the gist is the only difference between TA and 2x1 is only an induction. None of us disagree on rescue. The discrepancy is only an induction, and induction in a 2x1 cup model is two genes are required for induction versus the alternate hypothesis is that only one gene is used to induce the CI. And again, like these are very, very close. So I guess it's a positive thing if in a sense that we're so close now we have so much more that we agree on that we're debating these very nuanced things about induction. Needless to say, it's an interesting question mechanistically if you're interested in these mechanisms. And it looks like some systems, or at least one system, there's only one system that evidences the 2x1, which is the Drosophila melanogaster system. That system seems to work by 2x1, but many of the other systems appear to work by strict TA, where only one is inducing. And so that is the discrepancy that people are trying to resolve. And we might inhabit a world in which multiple models exist extant in the diverse insects around in the world. So these models are not mutually exclusive, and they might both coexist. But needless to say, this paper is debating what the case is for the CID system from WPIP. Here's why I really don't like the nomenclature that they're using in this system. CIF is so broad and vague, and we do use it as a broad term, but the CIF type system, you never know what they're talking about. They're specifically in this paper, they're specifically talking about the CID system from WPIP, which is the one that we cloned out and characterized. So that's the one they're talking about. And in our Nature micro paper, we doubled down on the fact that we thought it behaved as a TA system, not as a 2x1 system. And again, if you want to watch that nuance, go to the CI stories lecture. I'll probably put a link for it up here. If you haven't seen that, you're kind of ill prepared to watch this lecture. Needless to say, they're specifically answering this question of this CID system in mosquitoes, which is why this is interesting, especially to me, because I got the CID ring. Yeah, so okay, this is good. This is good to see. So just running through the introduction here, this is good to see from Flaminia's perspective, from Flaminia's expertise. she's saying Wolbachia does not appear to form stable endosymbiosis with Anopheles species. So again, I talked about before, there has been some quirks with trying to apply Wolbachia techniques to Anopheles. Here's her words on that. Only one artificial Wolbachia infection has been achieved in the germline of an Anopheles species. Moreover, upon endosymbiosis of Anopheles stephensi with W. alb strain, only partial rescue of CI and limited capacity for population invasion were observed. Interesting. All right, so that's the gist of the introduction. All right, so she's saying we chose to use the type 1 CIF genes, CIF-A, CIF-B, from the Wolbachia strain WPIP. Something smells siffy, so let me translate this for you. Type 1 CIFs are equal to the CidA B operon from WPIP. Very nice. She does cite the other nomenclature. That's kind. Thank you. So let's talk a little bit about transgenics. One of the reasons the research has progressed so fast in Drosophila is because the transgenic systems in Drosophila are just so robust. You can essentially pick and choose any promoter you want with this neat, tricky Gal4 UAS system. And so research on CI and Drosophila has really like shot forward. But everything in terms of transgenics, research lags behind a little bit on Drosophila in mosquitoes. It lags behind a little bit because the transgenics are much more complicated. There are Gal4 UAS systems in mosquitoes, but usually when people are doing transgenics in mosquitoes, they're kind of always like building their own constructs. And so here's a discussion of the promoter that they're using. This zero population growth promoter, ZPG promoter, which drives germline expression in both male and female in Anopheles gambiae. So they got this ZPG promoter and they're building this, right? So they're putting the ZPG promoter in front of, I imagine, the operon. Nope, they're splitting it apart. So they're putting this ZPG promoter, which is a germline promoter from Anopheles gambiae, in front of CidA or CidB. So this is the gist of their constructs. Interesting. So here's an interesting result. This is fascinating. Wow, I'm gonna cry. This is fascinating. It's always nice to see when there's so many fucking doubters out there. It's always nice to see people replicate your results in a totally different system. This is amazing. So look at this. Look at what they're saying. Co-injection of CIF-A. So okay, essentially like they're trying what we tried originally in Drosophila with this setup. In Drosophila, what we tried to make the individual transgene insertions. We tried to do an insertion of CidA, and we also tried to do an insertion of CidB. Okay, and this is good because this goes into the nuance of the models. When we were doing this in Drosophila, we tried to make both these constructs. Okay, we tried to do both of these insertions. This one worked, check. This one did not work after like multiple injections. And the reason that we thought that this did not work, it made total sense because CidB was a toxin, right? That makes total sense. So we thought this strengthens our hypothesis that this is a TA system because if you express the toxin by itself in Drosophila, it'll kill the Drosophila. You can't make a transgenic Drosophila insert for this gene that we were trying. That was our hypothesis. And so what did work was we made a construct that had both the antidote and the toxin, okay? And when we inserted this construct, it worked. And our hypothesis and philosophy of why we thought that this worked was we thought that the CidA attenuated the toxicity or weakened the toxicity of the toxin CidB, which again was evidence for the TA system. This was our whole point. That was one of the big points to our Nature Microbiology paper. Okay, so they started exactly where we started in Drosophila. They started in mosquitoes. Okay, so they're trying to make individual inserts of CidA and CidB. And what they say is They never got any that expressed CIFB only. This is important. This is a replication of our work in Drosophila. They're not getting any transgenic inserts of CidB only. Okay. They co-inject the two constructs, and what they got is they were able to get CidA alone, check, just like us. And they were able to get CidA plus CidB, check, just like us. But they never got a CidB by itself. And I assume from their abstract that their assessment is that that was because the A was attenuating the effect of B, just as we thought. So this is a good replicate of that experimental observation in a totally different system. That's amazing. And they're saying here, in all crosses, when females mated to the A and B males, they showed a striking degree of infertility. That's exactly what we observed. So when you put the two together, you get this infertility. The system turns on. And this was, again, people don't understand this nuance. The Bordenstein group interprets this as evidence for the 2x1. The reciprocal interpretation of this, which it looks like Flaminia and our group and Hochstrasser's group hold, is that it looks like when you put the two together, CidA is essentially attenuating CidB's toxicity, allowing you to actually get the transgenic insert, and then CidB is then effective enough to induce CI. All right, let's look at the figure. Let's look at the data. Here's their ZPG promoters. They got LOX-P sites to cut it out, it looks like. And they got some 3' UTRs, which I assume is for localization. It looks like they've got synonymous constructs here with alternate promoters, VASA, which is another germline promoter. So here's constructs. Here's the CI data. Wild type cross, that's high, 100%. Nice. So here's female, if you can't see this. Here's males. So none of these should be CI crosses. Here's wild type, no effect. Here's A in female. No, not really an effect. I bet it's not statistically significant. And then here's A plus B in a female. It does look like you're seeing a little bit of toxicity when you add the B. But these are not CI crosses. These are not CI. This is transgenes in the female. Here is the CI test. This is the CI test. The ZPG is the male germline promoter, I'm assuming. So this is the promoter they're using to turn it on in the male. Huge infertility when you have them both. So this is suggesting that the B is, what is this suggesting? I mean, I think what you conservatively conclude from this CI test is that these genes are involved in CI. A and B are involved, and you're getting CI, but you can't conclude which one is inducing it or which one is not inducing it from this data. And then here's a test of rescue where they're crossing the induced CI to the A, and you're not getting rescue. This is exactly what we saw in the fruit flies with this system. Not getting rescue. And our hypothesis at that time was it was not enough expression of A in the female germline to get that rescue. That was our hypothesis. And here's a second rescue test where they're doing an AB cross to an AB. And you can sometimes get this. I've seen like a weak rescue effect from crossing an AB to an AB in Drosophila. So it's good that they tested this. But essentially, they're not getting rescue. So essentially, the conclusion from this data is A and B can work in mosquitoes to induce CI. Rescue isn't working. And individual B insertion is not working. So I assume what's coming next is promoter tweaks. I assume that's what's coming next. Here they do the cytology. I'm not convinced by this stuff. Like, you see that this is the most common thing to do. I'm convinced by getting cytology at the first cellular division. like that one zoomed up super close. That's what I like to see. You see that in Sullivan's work. Of course, you see that in my work. I'm not as impressed by like seeing these. And one reason I'm not is because you can sometimes see these bridges in wild type embryos. Anyway, I don't bother so much with the cytology anymore looking at it. Yep, it looks like high expression levels. All right, so here's what's next. Just like I said, probably what's coming next is promoter tweaks. So they're saying high expression levels of CypA in females, now rescue. So now they're going to try to fix rescue. Try to fix rescue is section 2 of this paper. Exactly what we thought. Those fucking reviewers. Promoter dependent, yes. And Shropshire did good work on that. So they switched to the VASA promoter. Okay, so I guess VASA is a stronger promoter. So in terms of promotion, VASA is greater than ZPG. So I assume they're going to start tweaking now, promoters. And this is usually the trick to fixing rescue, according to the literature. All right, so let's take a look at these data. Okay, so this is QRT PCR. So they're quantitatively measuring transcript levels in A, and they're showing you exactly what I just wrote. VASA is greater than ZPG for CypA. So they're getting better expression with VASA. They validate that. That's nice. And then they try the rescue tests again, I assume. Control looks good. CI looks good. Rescue with weak promoter. No good, not working. As before. Rescue with strong promoter equals good. Very nice. This is very substantial now. That when you see these weak rescues, you push the expression. It's like there's like multiple examples of this now. Okay. So now what do we conclude after figure 2? A and B can induce CI in skeets. A can rescue. Very nice. Now I imagine figure 3, guessing is mechanistic stuff. What you would want to do now is you would want to weaken expression of B, try to get it in a male. Let's see if they do that next. Such a nice paper when you know what's coming next and then it comes. Very nice. Expression of CypB alone in males induces CI. So ZPG colony did not work, died or something. They were able to get F1 ZPG CypB males. So that means I'm presuming what that means is you do the microinjection. The microinjection also has a fluorescent insert. Do they have that in their constructs? So they're either yellow or red. Their constructs are also inserting these fluorophores. So they're either going to be yellow or red. And the F1s are going to be, I assume, the ones you microinject that survive the hatch, that pop out. And if they're yellow or red, they got the transgenic B gene. So then they test these F1s. We found that ZPG CypB males induce high infertility when mated to wild type females. All right, let's dissect Fig 3. Control looks good. A, no toxicity. I'm assuming these are the F1s they're talking about. Here's the CI induction with the 2 genes. Looks good. And here's induction of CI with just the B in the F1s. So the conclusion of this is that B is sufficient for CI induction. And the subconclusion, or therefore, 2 by 1 cup is invalid. In, I'll be careful, WPIP. Pip. CidB. Something smells siffy about this 2 by 1. Now, what the hell is this? Look at the legend. Here's the rescue test. So they're inducing, oh God, this must have been a pain. I feel sorry for you, Flaminia. All those F1s, all those micro injections. Oh, good God. Give that person a raise, please. But very nice. Very nice. That's stupid. I should cut that out. But this is very nice. Why do they have this twice? What is Z and V? Is this like a second replicate? I don't know. I don't know what this is. I don't know what this is, but it looks like it's the same experiment, unless I'm like reading something wrong. I don't know if they just like did it twice. Maybe reviewers were like, you must do this twice, or if this is a third time, I don't know. But it looks like they have extra replicates. If I got this wrong, then say something. But it looks like this is just like an extra replicate of the same experiment of F1s with CidB inducing CI. So again, just reiterating, I guess, B is sufficient for CI induction. And then here's rescue. Here's the rescue of CI. So the conclusion here is A rescues, B induces, which equals TA model. Here's strong A with weaker B, attenuation equals TA model. That's very good too. Here's some control. QRT, PCR control. So there's a lot of evidence for TA model, not just a little. And that's it. CIFB, yeah. Let's look at and then there's some other stuff where they see some effects in females. And they have some nice microscopy of the ovaries, which I have seen. I have seen some effects in females in some different operons. So this will be interesting. I'll give this some citations in our next paper. It's good discussion. Let's see if we can pull anything interesting from the discussion. So again, the big conclusion is these things apply. They're cross-applicable in Gambier. That's very nice. Bordency. What's with that patent? What's going on with that thing? Hochstrasser, what's with that patent? What's going on with that thing? Email me. Because it's now more valuable, regardless of how much we hate each other. Very nice, she cites me. Consistent with our own difficulties. I just want to say thank you, because it's so nice when somebody else actually understands the point that you're making rather than just disregarding you. Thank you. I like this person. Our findings are in contrast with results reported for CI in Drosophila melanogaster, where both CIF-A and CIF-B from WML were required to induce CI. What they're saying is their results are in contrast to the 2x1 cup model. Yeah, that was the line that I said. It looks like Bottlestein's got a few other competitors now. Oh. This is right at the page break. Let me try to reassemble this. So there are other studies which got toxicity from bees alone. And their ridiculous claim was that was not CI because they couldn't rescue it. That is a ridiculous claim. And here there's a discussion of those papers. Neither study was able to demonstrate rescue of these effects and thus could not conclude they were CI related. So this is a ridiculous claim that was made in those studies, that you can't conclude that they're related to CI, despite the fact that they're fucking CIF genes. Despite the fact that they're CIF-B genes, they concluded that the toxic effects were not related to CI because they couldn't rescue them. I thought that was the most ridiculous thing in the planet because they were just pushing death so far that they couldn't rescue it. They were choosing to make that conclusion because it invalidated their 2x1 model. So they disregarded that data and formulated this concocted conclusion to help keep their 2x1 cup model alive. Then when they paired the genes, CIF-A with CIF-B, very little embryonic lethality was observed, meaning that A was attenuating B, which is what I was arguing in the first fucking paper. So if you want to understand the ridiculousness to which people will bend their conclusions to their ridiculous models, read these papers, read this paragraph, which I'm not saying it's in this paper. I'm not saying read this paragraph and these papers until you understand what I'm saying, because that's fucking frustrating. When people are choosing to bend the data to their weak interpretation to keep their model alive. I have high blood pressure. And then the follow-up point from Flaminia from this Harvard paper is this point gets resolved if you tweak the expression levels, which is what I've been saying all along. If you kill things beyond death, you stab them so many times, they go a point beyond rescue where they cannot be rescued. So you have to weaken that CI induction. Rescue is very finicky, and you have to weaken to the point where the stoichiometries match out. That's what it seems like is evidenced in the data. Okay. Very nice paper. I like this paper, obviously. Okay, obviously I like this paper. I do think what has to happen next is I do think I have to go through the Shropshire papers. I have to get my mind right for that because I have to be in a kind mood. I do think that's what has to come next in this CI playlist. What I will say is, Nietzsche says, I don't want to be just a constant naysayer. I'm not gonna make movies trashing people's papers. I'm not gonna do that. So just know I'm not gonna make a movie that trashes people's papers. I'm only gonna do a complimentary movie. My worst insult is ignoring you. That's my worst insult. So if a paper's bad or I think it's bad, I'm not gonna make a movie trashing it. But I do think in order to get the roundness, like right now in this playlist, there's really only like half the side. I do want to get the roundness, and I want to try to present the rounded out argument. It's not going to be unbiased. Like I can't present it unbiased. Like I can't because I'm so deeply involved in the research. But I'm going to try to do it generously while pointing out my problems with conclusions from some of those papers. I like this paper a lot. Great job to Flaminia's group. Great job. I can't help but wish we could have worked together. You should have put the word in for the S, Professor. All right, bye-bye.
I am Worf, son of Mo. I have much to teach you about women. Notice how the Klingon lures a mate. Klingons do not pursue relationships. They conquer that which they desire. Today, I am a warrior. I must show you my heart. I travel the river of blood. I am a Klingon. Doubted. A demonstration can be arranged. That is how the Klingon lures a mate. Klingons do not pursue relationships. They conquer that which they desire. Today, I'm a warrior. I must show you my heart. I travel the river of blood. I am a Klingon. A demonstration can be arranged. I am a Klingon. A demonstration can be arranged. I am a Klingon. A demonstration can be arranged. I am a Klingon. A demonstration can be arranged. Earth females are too fragile. It is a good day to die. It is a good day to die. Show you my heart. I travel the river of blood.